Welcome to The Explainer. I am absolutely thrilled to have you with us today. We are going to explore a truly fascinating breakthrough in regenerative medicine, doing a complete slide-by-slide summary of the engineering kidney regeneration research presentation. I mean, just imagine taking a pig kidney, completely stripping away its cellular identity and transforming what's left into this smart, injectable hydrogel, a gel specifically designed to deliver human cells right where they're needed to heal. It sounds totally like science fiction, right? But as we're about to see, this is the literal cutting edge of tissue engineering.
Okay, let's dive into this. Here's our roadmap. We'll start with the cell delivery problem, look at the porcine blueprint, explore the physics of hydrogels, validate the biology, review the diagnostic metrics, and finish up with the real-world operating room challenges.
Starting with Section 1, the cell delivery problem, why injections fail.
Now, to really appreciate the solution here, we first have to understand the problem. Simply injecting stem cells into a patient without any supporting structure is, well, it's basically like trying to build a house without a frame. It just collapses. Cells desperately need a complex structural frame to nestle into. Without that precise framework, they can't anchor themselves, they can't function, and they definitely cannot regenerate complex tissues like a working kidney.
So, this missing structural framework, it's called the extracellular matrix, or ECM for short. You can think of the ECM as the natural, three-dimensional biological scaffold of an organ. And it doesn't just hold cells in place like a kind of biological glue, it actually provides vital biochemical instructions. It literally tells the surrounding cells how to behave, how to grow, and how to differentiate. Honestly, recreating this matrix is the holy grail of tissue engineering.
And the stakes here are incredibly high. When naked cells are injected directly into a host, they scatter, they completely fail to anchor, and ultimately, they just die. The survival rate is exceptionally poor. But when cells are safely anchored in their native extracellular matrix framework, they thrive, they survive, and they actually execute their necessary biological functions. The scaffold is every bit as vital as the cell itself.
Moving on to Section 2, engineering the porcine blueprint, creating the ghost organ.
So, if humans need this perfect extracellular matrix to heal, where do we get it? We can't exactly build it easily from scratch in the lab. So scientists look to nature, specifically to the porcine, or pig kidney, to act as the ultimate structural blueprint. It's a four-step process. First, you harvest a raw porcine kidney. Second, the specific cortex tissue is isolated. Step 3 is where the magic happens, the decellularization. This strips away all the pig cells, leaving behind this wild, white, ghost-like skeleton of pure protein. And finally, in step 4, that skeleton is engineered into a smart, injectable hydrogel customized specifically for human cell delivery.
But wait, how do you actually turn a piece of pig tissue into a safe scaffold for humans without triggering a massive immune system rejection? Well, it requires a really intense, multi-week chemical washing protocol. They use a 1% SDS detergent continuously for days, and that's followed by Triton X100 for another whole week. Then a massive 7-day PBS wash eliminates all those residual detergents. Finally, an overnight DNase treatment digests any tiny bits of leftover genetic material.
Now, what's really interesting here is just how effective this is. The data shows a massive drop in DNA levels after the wash. This protocol ruthlessly eliminates the cell nuclei and the DNA that would normally trigger an immune response. But crucially, it successfully preserves those vital architectural proteins like collagen and glycosaminoglycans that our human cells are going to need to survive. Once we have this completely clean matrix, we have to deliver it. So the decellularized matrix is freeze-dried into a fine powder. When it's mixed with a saline solution at a neutral pH of 7.4, which is a totally safe environment for cells, it remains a fluid, flowing liquid at room temperature. This is a game changer because it means a surgeon can easily load it into a syringe along with the human cells and just inject it. It only cross-links and hardens into a solid gel when it hits human body temperature at exactly 37 degrees Celsius.
Section 3, the physics of hydrogels, liquid versus solid.
Okay, we've talked about the biology, but for this to actually work in a real hospital setting, it really comes down to physical biomechanics. This new material has to perform a crazy paradoxical trick. It has to behave like a liquid during delivery, but act like a solid once it's inside the body. To understand this viscoelasticity, just imagine a bouncing ball. When a hydrogel acts like a solid, it stores energy. That's its elasticity, its storage modulus. But when it acts like a liquid, it loses energy, dissipating it into the ground. That's the loss modulus, or its viscosity. Testing actually shows this engineered hydrogel maintains a highly stable viscoelastic range. It handles physical stress perfectly without fracturing.
So the absolutely crucial point here is this delicate balance between states. If the matrix is too liquid, if it has way too much viscosity, the human cells you inject are just going to wash away in the patient's bloodstream. Useless, right? But if it's too solid, if it's purely elastic, doctors literally cannot physically push it through a syringe. The sheer utility of this material is that it dynamically transitions between these two states based purely on temperature.
But how does it stack up against what we already use in the medical field, like pure collagen type 1? Well, when warming up to 37 degrees body temperature, the engineered matrix cross-links and achieves a completely stable, hardened state much faster and far more robustly than standard collagen 1. And the internal architecture of this new hydrogel is vastly more complex and interwoven compared to the somewhat sparse structure of standard collagen.
Now, a successful scaffold also has to know when to, well, leave. It must be biodegradable to make room for the newly regenerated natural tissue, but it absolutely cannot collapse prematurely. The testing shows exceptional environmental stability here. Over 21 days, the hydrogel loses only 1.2% of its mass. This structural persistence means it degrades slowly enough to safely support massive amounts of new cell proliferation.
Let's move to Section 4, Biological Validation and Growth, Proof of Life.
We know the physics hold up, but what happens when we actually introduce human cells back into this ghost organ matrix? On day 1, in both standard collagen and our new engineered matrix, this all survived just fine. But fast forward to day 21. In standard collagen, growth is pretty sluggish, but encapsulated inside the porcine matrix, human kidney proximal tubule cells are visibly thriving. They're branching out, and they are robustly multiplying inside that rich architectural environment.
And here is a massive, massive biological insight. This hydrogel doesn't just passively keep the cells alive, it actively tells them what to do. Undifferentiated cells placed into this matrix show significantly higher and faster expression of specialized podocyte and tubule markers. The matrix itself is actively guiding the cells to differentiate and regenerate specialized kidney tissue. It's doing the communicating.
Section 5. The Ultimate Diagnostic Matrix.
When we pull all of this data together, we can see exactly why researchers are so incredibly optimistic about this specific tissue engineering pathway compared to older methods. Pure, naked cells, they fail completely at structural complexity, long-term viability, and differentiation. Standard collagen type 1 is okay, but it has low structural complexity and super slow gelation. But the engineered hydrogel, it gets green plus signs across the entire board. It's highly injectable, extremely complex, gels fast at body temperature, ensures high cell viability, and actively signals tissue differentiation. It is just an outright winner. Which brings us to Section 6, Bridging Lab and Clinic, the Operating Room Challenge. Of course, science in a pristine, sterile lab is very different from a chaotic, time-sensitive surgical theater. We mentioned earlier that the gel crosslinks automatically when incubated at 37 degrees Celsius. Well, there's a catch. That process takes 45 minutes to fully stabilize. In an active operating room, with a patient literally open on the table, surgeons simply cannot sit around waiting 45 minutes for an injection to harden. That is a massive clinical roadblock that has to be solved. During the really fascinating academic Q&A session on this research, top experts fiercely debated solutions to this. Some suggested engineering the matrix to gel at lower room temperatures, like 25 or even 15 degrees. Others proposed utilizing a centrifuge at 1,000 RPMs for immediate mechanical hardening right there in the OR. Another brilliant idea was adding chemical triggers, like alginate, which is derived from algae, which instantly cross-links upon injection, completely bypassing that temperature delay.
Which brings us to our clinical trajectory. We have a smart scaffold that removes immune triggers, we have thermoresponsive delivery, and we have vastly superior biological regeneration. I'll leave you with this provocative thought. If bioengineers can just beat the clock and solve that 45-minute operating room delay, could this ghost organ matrix become the ultimate definitive cure for chronic kidney disease in our lifetime? Thank you so much for learning with us on The Explainer, and keep your curiosity focused.